Velkomin. Velkomin á Málstofu Hafrannsóknastofnunar. Í dag erum við svo heppin að fá hérna erlendan gest til þess að halda fyrirlestur. Sorry. I'll switch to English now. So Davina Derous is a guest here at the Marine Freshwater Research Institute, working on whales. I have not the total information on what she's doing, but I hope she will give the information during the talk. Davina is currently a lecturer in environmental physiology at the University of Aberdeen in Scotland. She has a bachelor's degree in dietetics and nutrition from Waves University for Applied Sciences. That's in Belgium. She did a master's degree in nutrition and health, specializing in nutritional physiology from Wageningen University, and that's the Netherlands. In 2013, she started her PhD at the University of Aberdeen, where she focused on how changes in food availability can impact changes on a molecular level in different tissues. She continued this work during her postdoc at the University of Aberdeen, where she is currently, as I told you earlier, a lecturer. Now, in 2019, she established a research lab and now focuses on how changes in the environment can impact metabolism by using molecular assays and comparative physiology approaches in several species. And I think that's related to her talk, if I'm not mistaken. Davina, please.
Thank you for the introduction. So I have a slide at the end of my presentation that will talk a little bit more about what I'm actually doing here and what I've been doing for the last week. So just a quick introduction to the team. So we have Joe Kershaw, who is a marine mammal scientist and also a lecturer at the University of Aberdeen. And then I have two PhD students, which are Alexandra and Devin, and they started their PhD in September 2022. So I'll talk a little bit more the work I'm doing related to finding novel health markers, and it's always good to place this kind of things in a framework. And the framework I work in is population consequences of multiple stressors. So this is the graphical presentation of that. So basically what I focus on is how stressors can impact population dynamics. And as you can see here, for example, stressors, for example, foraging disruption, but also underwater noise and vessel noise, contaminants and things like that, can have an impact on the physiology of whales and dolphins. And then any changes in the physiology will also be reflected in changes in health. And then, for example, if we see a decline in health, we also see change in survival and reproduction. So if we have less animals being born, then we also will have a decline in, for example, our population dynamics. So there's different ways to measure health, and there's a few listed here. And my specific focus is on how our metabolism, energy stores, and how we can find novel markers related to how much energy is available, for example, to support survival and reproduction. So there's a very small link between the amount of fat we have. So if we have plenty of fat available, then we're able to invest what we call energy in survival and creating offspring. And when we don't have a lot of fat available, then mechanisms such as reproduction will be the first ones that start shutting down. So with less fat available, we also see less offspring being born. So the first question that I'm going to ask is, well, let's have a look at the current health markers. So the current health markers in cetaceans are blubber thickness. So this is just this really thick fat layer right under the skin, and you can see an example here from a stranding, and then here you can see an example of a biopsy. So biopsy is usually taken when you're on a boat in free-ranging animals, and this is the little sample you get out when the arrow bounces off the skin and comes out. So what I did together with the Scottish Marine Animal Stranding Scheme is look at historic data from 1991 for cetaceans. And then we had different species in there, so they ranged from harbor porpoises all the way up to sperm whales. And we had 3 different positions of blubber thickness, so this was dorsal, ventral, and lateral. And then we also looked at the causes of death, and this is what we used as our HELT. So we had species that died of trauma, so quite often these are quite healthy. So trauma, for example, is vessel collision we had, or for example, also bycatch. Infectious diseases is when there was a very clear sign that the animal was diseased, and other could be anything from contaminant from plastic in the stomach to other that we just don't know what they died of. But quite often, when they're healthy, we'll have to give the label healthy to them as well. So what we found was a very poor relationship between health and blubber thickness. So I need to stress that we're not the only people that found that. So there's a lot of papers out there that shows there's a very poor link between health and blubber thickness. And that's mainly because blubber has a huge amount of different functions as well. So just narrowing it down to health and blubber thickness is a little bit too simple. So the big question is, well, how do we go and find novel health markers? So what I do is I use novel techniques. So I focus on what we call the omics, which I'll explain later on what that is. And I also take a system-wide approach. So with the system-wide approach, I'll just explain a little bit what I mean with that. So we have certain stressors, and then that stressors can have an impact on, for example, our gene expression. So we know that certain genes will be either upregulated or downregulated, related to a certain exposure. And then that gene expression will drive changes in our physiology. So think, for example, there will be change in hormones that will be signaled, or certain enzymes that will be switched on and off. And that will have an impact, for example, that we start breaking down our fat, or, for example, we start building up our fat, depending on what the stressor is. And then that physiology can have an impact on our behavior. So if you start putting all these different puzzle pieces together, what we can create is a certain phenotype. And this phenotype will then be, for example, diseased or healthy, and then all these different aspects, what we call system-wide approach, will determine how that phenotype looks like. So first of all, what we need to do, and especially in cetaceans, what is really difficult to do is these are free-ranging species, so they're exposed to lots of different things. So if you want to start understanding the association between health and their metabolism, what we first need is a very controlled environment. So this is what I did with bottlenose dolphins. So we did a fasting experiment, and the reason that we did this was to mimic foraging disruption. So this was together with the US Navy Marine Mammal Program. So they have about 80 bottlenose dolphins that they keep in their facility, and they are trained for various tasks, and a subset of them we can also do very non-invasive experiments on them. So what we did was a 24-hour fasting, and we took blood samples pre and post. So I just highlighted blood samples here because I've been talking about fats, so we're switching now to blood. So the reason why we take blood is connected to all different organs, so it gives us a really good indication what's happening in the overall body, not just in one specific tissue. And what we did is we did metabolomic identification. So I'll just quickly explain what metabolomics are. So we have our DNA or genomics, and that gets transcribed into RNA, and then RNA gets translated into proteins. So proteins are our hormones and our enzymes, and that goes through our entire body to our entire metabolism, and then the end product, what we have, is metabolites. So metabolomics is that snapshot of what's happening at the end product of our metabolism. And this is the result. So this is a principal component analysis. So what this shows is the variation that we see in that metabolomic fingerprint. So we have the post in blue and the pre, so the pre-fasting in orange and the post-fasting fingerprint in blue. So as you can see on the bottom here, we see very nice separation between the two groups, and that indicates that 52% of the variance is explained by that separation. So that means that if we see any changes in our metabolite, either up and down, that 52% of that variation is explained by exposing them to the diet, to the fasting. So that means that we can expect quite a large change when we start comparing exactly which metabolites are different pre and post fasting. So that's the next step we did. So we compared which metabolites were changed after fasting. And this is the plot you can see here. So on the x-axis you have the log twofold change. So that just means that we overlaid the pre versus fasting, and then, for example, if we see a positive change, which is a positive logfold change, it means that metabolite will be higher in the fasting, and if you have a negative value, it means that metabolite will be lower in the fasting. And you can see on the y-axis you have your p-value, so the higher the metabolites are on the y-axis, the more significant they are. And we also color them according to their role in metabolism. So what you can see based on the colors, the positive metabolites were mostly blue, and the negative ones were mostly green and orange. So were amino acids and proteins. So it means that they are releasing lipids into their blood, and they are actually taking up amino acids and proteins from the blood. So what does that mean? So that shows an immediate switch to lipid usage, and I'll explain a little bit what we expect to see. So that's a very stark contrast for what we know from model species. Model species, I mean humans and mice models. So most of the fasting experiments that we know is based on those species. So what we see first of all is we see a decline in glucose. It means that the animals start using up the glucose first. So that's stored in, for example, the diet, but also your liver in the form of glycogen and your muscle. And then only after a day or 2, 3, what we see is what we call the phase 3. We see that change into lipids, use of lipids. So what we see in the dolphins that are fasted, that within 24 hours, we actually see an immediate switch to lipid usage. So they seem to be not using any of the stored glucose in their muscles or the liver at all. What we also see is that there's less amino acids circulating. So as you can see, this usually happens around day 2 or 3 in the model species. So within 24 hours already, we see that dolphins are starting to break down their muscles actively to start using up the amino acids. So what does that mean? So it means that what they're actively trying to do is actually increase their glucose. And there's also previous studies that shown that when you fast dolphins, for example, for 72 hours, there's an increase in glucose level. So it looks like after 24 hours, they're already starting to break down their amino acids, and they're trying to convert that into glucose. And that's what we call gluconeogenesis. Now, the big question is, why do they do that? So if you think of the diet of a dolphin, we know it's very devoid of carbohydrates, very rich in proteins and fat. And we know that blood and brain, for example, are very specific tissues that can only use glucose as an energy source. So for example, our fats, our liver, our muscles, they can also use lipids as an energy source. But our brain and our blood are the only ones that can use glucose specific. So there's 2 things associated with that. First of all, they need energy for the brain. And second of all, the brain also needs to produce heat. So the dolphins are in the ocean, they can get cold, and they need to keep their brains warm. And that's why we think that breaking down those amino acids for producing more glucose is to keep their brains warm, and also to provide their brain with energy, basically. So this can cause some implications for multiple stressors. So what do I mean with that? So for example, if you have a dolphin that's fasted or does not have a lot of food available, and then we add an extra stressor to that, so for example, we introduce vessel noise to it, there might be questions related to, well, if they're already starting to use up their muscle, what will happen if they have that extra stressor? Can they actually do their fight and flight reaction on top of that as well? So, okay, so now we know that we can apply metabolomics to a very controlled environment, and we can get some information out of it. So the next question is, can we actually also use this in wild populations? So the next bit of data I'll talk about is a project that we did with, again, bottlenose dolphins in the Charleston Harbor, South Carolina, and also the Indian River Lagoon in Florida. And we had animals that were either healthy or diseased, so they had a physical examination, and we classified them according to these 2 different groups based on what the vets classified as healthy and unhealthy, and also based on a blood paneling that they did. So they looked at markers for disease in the blood itself. So again, we collected blood, so we also did blood chemistry and metabolomics. So just as a brief reminder again, so metabolomics is that end product of metabolism, and is that snapshot of what's happening. And this is the result of the blood. So we can see on top here, these are different hormones that are associated with stress. So in the Indian River Lagoon one, which is on the bottom, we can see the red one are the diseased ones, and the blue ones are the normal ones. So we see some indication that stress is elevated, but it's not significant. But however, if we look at the Charleston Harbour ones, we see that the disease ones do have higher stress levels, which was significant. Very interestingly as well, we see differences between the different populations. So how is stress regulated? So we have a certain stressor that will signal specifically to the bit in our brain, which is called the hypothalamus, and that will send a signal to our pituitary gland, and then that will release one of the first stress hormones, which is ACTH, and that will go to our adrenal gland, and our adrenal gland will, in response, secrete our cortisol and our aldosterone, and that will have an effect on our metabolism. So one of the first things that will happen is, again, that gluconeogenesis. So again, it will signal to the liver to produce more glucose. So that is to make sure that we can do our fight or flight reaction. So it will just produce lots of energy so we can either decide to fight or flight. Second, it will do that breakdown of protein that is necessary for gluconeogenesis. And a third one that will do as well as free fatty acids as well. So there's two things that happen. So amino acids for gluconeogenesis and fatty acids for energy availability for the other tissues. So this is data from stranded bottlenose dolphins, and this is the adrenal gland that we just sliced in the middle. And this paper really nicely showed that those dolphins that were exposed to acute stress, you can see that the inner band of the adrenal gland doesn't have much morphological changes. But if you see those dolphins that have chronic stress, you can see that when we slice the adrenal gland in the middle, you can see there's already chronic changes to the actual structure of the organ itself. Okay, so just a little side tour of how stress is regulated. So we'll have a little look now at the metabolic profile. So these are all the metabolites listed here, and blue means lower expression and red means higher expression. And then you can see the CHH, diseased and normal dolphins up there, and then the same for Indian River Lagoon. So what immediately pops out here, as you can see, is that the dolphins are not really clustering together according to their diseased or health status. So what we do see is that they're more grouping together according to the location. So based on this plot, what we see is, especially these metabolites, we see that the Charleston Harbour dolphins have a different profile compared to the Indian River Lagoon dolphins. So then again, we did that principal component analysis, and as you can see, if you look on this axis, 33% of the variation explains the different grouping, but only about 7% actually explains any differences between the disease and the normal state. So it shows that there's not overall, if you look at the overall metabolic profile, there's not much difference between the diseased and healthy species. So next question we asked is, well, is there any specific metabolites that we can then pinpoint exactly what's different between the diseased and normal ones? And this is the result here. So we've got your Charleston again and your Indian River Lagoon there. So you can see of the 500 plus metabolites, about 20 were significantly different, and this was the same for the Indian River Lagoon and Charleston. So, and what is most importantly, when you're dealing with, for example, metabolites, a lot of these metabolites will be unknown, and that's because we don't have a good reference for bottlenose dolphins. So lots of these metabolites we can't identify because we just don't know what they are yet. So it needs a bit more of work to pinpoint exactly what they are. And these are the ones that are different. So you can see that both of the different locations, so it's some overlap. So we see that, for example, especially amino acids are again down. And if you look specifically to Charleston and Indian River Lagoon, as you can see, most of the, again, are amino acids. So it's a bit the same story of our fasting story. So what we see again in these animals is that when they're in poorer health, amino acids seems to be again the story that pops out. So what we also saw was that the diseased animals have a higher stress level. But then the big question that we have to ask here, which I brought up with the fasting example, is it because the diseased animals actually have more stress, or is it because they actually experience stress differently? So these animals were live captured, so they were hold a net and then blood was taken. So for them, that's an extra stress. So it could be that the health individuals were able to cope with the extra stress and were absolutely fine, while the diseased ones couldn't cope with the extra stressor on top of it. So, and that's why we see these metabolic changes. So it's a bit question again, why do they have this higher stress level? So as I mentioned again, we again see circulating amino acids. So again, that shows breakdown of the muscle. So it looks like the diseased individuals are also starting to break down their muscle for extra energy. So quite likely, again, this is for the new formation of glucose to fuel their brain, for example. So what we do know is that the diseased animals, although we don't really see a massive difference in the overall metabolic state, we did see very specific metabolites that were changed. So to conclude this, again, what is the impact for conservation? Will they be able to cope with any added extra stressors to them if they're diseased, yes or no? Do we have to start changing policies of how species deal with different stressors based on these results? It's a bit of a big question mark at the moment. So another study we like to talk about is what Jo Korsha did during her PhD. So Jo focused on finding novel biomarkers related to body condition, and this was in collaboration with the Michigan Iceland cetacean study. So, and the big questions she asked was if we would be able to use proteomics and blubber to just see what the function of blubber is. And this was in Canada. Sorry, just a quick recap of what proteomics is. So proteomics is a step before metabolites. So it's a step after your genes. So your genes, for example, regulate certain proteins, for example, hormones, certain enzymes, and things like that. So Jo had minke whales, about 10 all females, and the data was collected in 2014 from Canada. And the question she asked is, first of all, can we use blubber to look at the protein expression levels? So what she then did is she mapped proteins to known biological pathways. And I'll just quickly explain how we do this. So proteins are identified and get a certain intensity value associated with them. So that's based if a protein is high expressed or low expressed. And then what we can do is we can order that protein according to the intensity value itself. And then, for example, if this is a certain biological pathway, and this is a certain biological pathway, and this is another one, and these are all individual proteins, what we can do then is take our 5 proteins here, and we can overlap that with all the biological processes. And then we can give them a certain overlap score. So for example, if you look at pathway 1, we'll only have 1 protein associated with these 4 proteins, and then for example, pathway 2 will have 1 of 7, and then pathway 3 will have 3 of 5. And those two things together, if a protein is higher expressed or lower expressed, and the overlap of how much is mapping to the known biological pathway, is what we call an enrichment score. And that enrichment score will tell us specifically if a certain biological process is higher expressed in for example, these baleen whales. So this is just results. So these are all the different proteins and in their biological pathways that are interacting with each other. And the main take-home message here is that we see lots of immune regulation in the blubber. We also see some, for example, O2 and CO2 exchange, which is not surprising because blubber has, for example, lots of arteries running through it to provide oxygen to the tissue. So we see also some metabolism popping up as well. So this is a really good starting point to start targeting specific proteins associated with, for example, baleen whales. So summary, we're not there yet. We're still far away. So we know now that we're able to get metabolic fingerprints from free-ranging cetaceans, but we need more mechanistic work to really start disentangling exactly how health and metabolism is connected with each other. So how do we start doing that? So how do we start unraveling how health and disease, for example, is related to their metabolism, and how do we start finding novel markers specifically associated with that? So the first thing that we have is we have an issue of information. So how do we deal with unknowns? So we know that free-ranging cetaceans are exposed to lots of different things in their environment. So this is a really nice paper that described all the different exposures in the water. So as you can see, based on this figure, there's lots of unknowns that we just don't know when we take a sample of a whale or a dolphin, what exactly they've been exposed to. Second is their life history. So quite often when you take a biopsy sample, you don't know in what, for example, reproduction stage they are, if they're lactating, if they have a calf, yes or no, have they recently been lactating, for example, or what age they are quite often as well. And then third, what we're dealing with is a snapshot. And I refer back to this tiny plug of blubber that we're dealing with. So we know that our body does not regulate things in unison. For example, we know that our liver and our brain, our muscles are all closely working together. So it's quite hard to look at that one snapshot that we get in blubber to make to conclude exactly what's going on related to their health. So this is the work I'm doing with the Scottish Marine Animal Stranding Scheme. So the advantage of that is that when you have a stranding like that, you get access to lots of different organs. So you can access for example, the blubber. You can access the muscle, liver, and brain. So it means that we're not limited to the amount of samples that we can collect. It also means that you can do a full investigative body necropsy. So you can look at, for example, the stomach content. You can see if it's recently been fed, yes or no. You can look at if it's diseased. Does it have, for example, parasites living in it? And you can really start that system-wide approach to try to link all of those different aspects together. So what are the different research techniques I use to try to answer those questions? So first of all, I look at the morphological measurements. So look at blubber thickness, body mass, girth and length. And then I also look at histology. So the example I give here is blubber, but I do this, for example, for muscle and liver as well. Specifically in blubber, we can ask about the number of fat cells. Do we see if this changes, for example, with fasting, yes or no? But we can also look at the size of it. And then the third bit is the molecular measurements I talked about. So the proteomics and the metabolomics. So this is a paper that Joe published a few years ago now, and this was the first proteomics in stranded cases. So we know that we can get proteomics out of stranded cases, which is really good, because quite often we don't know when exactly the animal died, how long the animal has been in the beach, will there be degradation of the tissue? So these are all questions that we're trying to ask with strandings, if stranding cases can be helpful to inform what we want to inform. So the answer to that is yes. So we know that we can get proteomics out of blubber tissue. So then what are the different research questions that we're trying to answer? So first of all, how do they actually store their energy? And does that change with age, sex, and body condition? And is that linked back to the gross pathology? So we also send samples off for pathology. So we can look, for example, if there's any specific diseases that's happening in the tissues. Then we can specifically in blubber, do we see any changes, for example, in the fat cells? And then the last question is, which biological mechanisms and pathways are changed? And then these three combined, we can start developing novel health markers that when we have that little snapshot of the blubber, we can have a really specific biomarker that tells us if this animal is healthy, yes or no, in free-ranging cetaceans. So I just wanted to give you some preliminary data I got back last week. So we had a very unique case where we had a beaked whale stranded in Scotland, and it was a mom that was lactating and a calf. So it was a live stranding, and then sadly they died. So the adult female seems to be in really good body condition, but the stomach contact showed it was not fed recently. She was also lactating, but sadly it was less than anticipated based on the size of the calf. And some of the pathology shows that there was some negative energy balance going on, and the fat and the liver and the fat looks like it's been deposited. So the liver gets this really weird color when there's a lot of fat in it. So just looking at it looks like there was lots of that fat deposition going on in the liver. So we did proteomics on both the calf and the mother, and we did blubber and we did the liver. I still need to analyze blubber, but I can tell you a little bit about the liver. So we got 855 proteins back from the liver. So we mapped those to known biological pathways just to have a bit of an idea of which kind of mechanisms is happening in the liver. And this is the result we get out of. So each of those little circles is a biological pathway, and then the link between them is how much they are regulating each other. So nothing special going on in the liver, so we've got our normal energy metabolism pathways popping up. What was really interesting is that we see urea metabolism popping up and catabolism as well. So urea is usually an indication, again, of the breakdown of amino acids. So we have some lipid metabolism here happening as well, and catabolism, again, was mostly related to amino acids. And then detoxification and oxidative stress. So that's a really interesting one because, for example, if animals are dealing with contaminants in their environment, these are the pathways that would be regulating as well. And then we had some really generic cell structure and transport going on. So again, compared to the bottlenose dolphin story, again, it looks like when they're in a negative energy balance, which you know she was because the stomach was empty, it looks like the amino acid pathways are starting to pop up. So quite likely, again, she was breaking down her muscle to provide that energy storage. So as an extra step, I mapped them to known diseases, and same story again. It looks like some known diseases was related to amino acid concentration. So I'm going to keep repeating it. So it looks like breakdown of muscle seems to be a very key point in cetaceans, and this actually goes inside with some of the stranding reports that we get as well, where we see that they can have a full layer of blubber, but we see that the tail end of their muscles actually are starting to waste away, basically. So why am I here? So long half an hour to talk about what I'm actually doing here. So we recently got a Leverhulme funding trust, so I'm collecting blubber samples from those in the coast of Scotland. So I just wanted to show you an overview of the species that we get in Scotland. So we mostly have dolphins and harbor porpoises. Those are the main ones. But what I'm doing here is I'm actually subsampling the two mass strandings that you had in Iceland. So I'm subsampling the pilot whales, the one of 2019 and the ones of 2021. So why specific pilot whales? So they're very sensitive to, for example, vessel noise and things like that. So quite often when they strand, they seem to be in a really good body condition. So compared to the ones in Scotland, so most of the species that strand in Scotland are more on the diseased end. So that's, and we also get less of the variety. For example, we get more of the harbor porpoises and the dolphins. So it would be really good from a comparative physiology approach to see what exactly is happening in the blubber of these pilot whales and see if we can find any markers specifically for pilot whales. And then what we'll do with them is we'll identify that metabolic fingerprint in the blubber. So just a big thank you to everyone that was involved in the different stranding projects, for example. So that's Málstofa as well, and then Philippa. And then we have the fasting experiment, everyone that worked on the bottlenose dolphins, and the nýjum project as well.
Thank you. We have time for questions. Einar?
Thanks. That was very interesting. I'm wondering about, so in the US experiment where you have the wild individuals and you have these two stressors, and one of the stressors is just for everyone, the net.
Yeah.
Do you think there's a way of quantifying that stressor? Like, so you get like the baseline, basically, what is the stress of someone that's not in a net?
Yeah, no, that's a really good one. So we thought about doing that. So the only way we can do it is, for example, I wish I had a video of that. So there's the marine, the ones of the Navy, I just can't call them the Navy dolphins. They did what's called an out-of-water stress test. So it kind of mimics the stress they have when they're, for example, captured. So they let them beach themselves, and then they measured cortisol, stuff like that. So I didn't do it, but we could have done is take those baseline values of them because we know they're used to being handled, and we could compare them to the net, see what exactly is the difference. And I think he actually did it. I think Patricia actually compared those data. So I think the paper is out there, but I'm not quite sure what exactly they compared. But yeah.
So you could, with that, do you think you could see like how really stressed are the ones that are deceased? Yeah.
Yeah.
Do you have a question? You are a question, right?
I have a question. Can I? Thank you, Einar. Just to clarify, so the non-lethal biopsy, that's not going to give you as much information, is it, as the stranded?
Sorry?
The non-lethal biopsy
Yeah.
to cats.
Yeah.
That's not going to give you as much information, is it?
Yeah, it's just going to give us a little snapshot, and quite often as well is we only get the top layer of the blubber as well.
Yeah.
So lots of studies have been done trying to figure out exactly, for example, what's happening through the different blubber layers. So we know that the fatty acid composition, lipid composition changes throughout the blubber layer. So, for example, the top one is believed to be more involved in structural support. Middle one, for example, more storage of the fats, and then the closest one, which is really close to the muscle and everything else, is believed to be more involved in metabolism and things like that. So it's a bit of a question, if we take that top layer, are we really going to find any changes related to, for example, health markers, yes or no?
So the reason I'm asking is that we are lucky these past days that we have a humpback whale here in the harbour. And we can identify it as an individual, actually. So his name is ISMN 1625. So we know the guy. So could you do a repeated biopsy, repeated snapshots? And for example, what I've been thinking for the past days is that this poor guy has been bullied by the whale watchers, basically.
Yeah. So that's actually the second part of the Liverpool project, which I didn't talk about. So we have samples from fin whales, and last one is, I'm blanking on that one, but we've got two different species, and we have them when they arrive on their feeding ground and when they leave on the feeding ground.
Yeah.
So we'll be able to compare them, and we also have several years as well. So you will be able to compare exactly time-wise what's happening with them.
Yeah.
So in theory, if you have markers in that top layer and you know that they are going to change with, for example, he might be really hungry now because not a lot of fish is left by the end of it, or he has built up his fat reserves enough, then we might be able to pinpoint some kind of...
Because in the beginning it was like 12 or 15 whales.
Yeah.
But now it seems to be one individual or maybe two.
Yeah.
All the others are gone, and they're not being bullied by the whale watchers. This one is singled out almost.
Yeah.
So I'm just thinking about the stress that he's experiencing.
So it could be, for example, and this is, it could be just lots of different reasons. So maybe because he might be on more energy deficient, that he might be sticking here more, while the others can actually invest their energy to go away and to go other feeding grounds.
Exactly,
So but these are things that we could, if you have the fat of them, we could have a little look at exactly what's happening in the blubber.
More questions?
Thank you for an interesting presentation. Brand new topic for me. But I was wondering, you were saying that you don't know how, if the whales kind of react to stressors like individuals, like human, like some of them would experience it very stressful, others not. So I was wondering, have any studies been done on that? And how do they recover from stress? Do some of them recover right away, or does it take a long time?
I don't know, actually. So we don't know how quickly they recover from stress, but some experiments with, for example, rats and mice, I know it takes up to like 3 weeks for them to fully recover, or 2 weeks to fully recover from the gluconeogenesis and breakdown of the muscles, and then that fight and flight response. So it takes a quite long time to come back out of that energy dip. From specifically to whales, I'm not sure it's been done. I know there's been work done on cows that looked on chronic stress, But specific to whales, I don't know. Yeah, I think people must have done some. There's a huge amount of work done on cortisol and things like that in different species. Yeah.
Thank you.
More question? Oh, you have one.
More like logistics. There was a pilot whale stranding very close by the airport here in 2018 or 19, and it was like 58 individuals, but there were 30 that were not saved or something. And are there no biopsies from those that you want to use, or is that, did we not?
Is that one of the? Yeah.
Oh, okay.
Yes.
Cool.
So we got that.
Okay.
Yeah.
I just
Thank
because
you,
it was
Tally.
northwest and northeast or something, or maybe I just read wrong. Okay.
Okay, good. Thank you.
Thank you.